Okay, so what we've got here is a model of the DNA double helix. Now, this is a really important structure in biology when we start thinking about the mutability as well as the inheritability of the DNA in terms of genetics. So let's go over those basic structures. Notice that this is a helical shape. It's a twisted staircase, if you will. And that twisted staircase is going to have nucleotides that make it up. Okay, now a nucleotide has three parts. Those three parts are the sugar, the phosphate, and the base. Okay, so the sugar, phosphate, and base make up one nucleotide. And there are four bases that we use in DNA: adenine, thymine, cytosine, and guanine. Now, when we go ahead and take a look at these, they bind together according to Chargaff's rule. So let's say this is adenine. Adenine is only going to bond to thymine on the other side. So adenine's always with thymine, and cytosine's always with guanine. Okay, now in terms of our structure, pyrimidine and purine, you always have one double ring base called a purine with one single ring base called a pyrimidine. So as we're going ahead and bonding together, remember that you always have A to T, adenine to thymine, and always have C to G, cytosine to guanine. That's according to Chargaff's rule. Now, again, nucleotide pieces, we've got a sugar, phosphate, and base, and the bases are connected together here with hydrogen bonds. Now, it may seem contradictory to go ahead and use what we typically consider one of the weaker bonds to hold together this incredibly important molecule, but there's a reason for it. First of all, there are millions of those hydrogen bonds, so their strength adds up over time. But the other reason is in terms of DNA replication and transcription. When we need to go ahead and make a copy of the DNA molecule, we need to do so by first unwinding and unzipping the double helix. So we start by unwinding it, okay, and as we unwind it, then we're going to unzip it and break these hydrogen bonds. Now, we don't want to leave them broken, so we're going to go ahead and put them back together. Now, if this was, say, a covalent bond, for example, that would be a really high energy need to go ahead and break and a high energy need to remake it. But with a hydrogen bond, it's not insurmountable. We can go ahead and break this bond when we need to, and then reform that bond when we go ahead and put the molecule back together. Okay, so hydrogen bonding is actually a really cool thing when you stop to think about it. We use a weaker bond, but millions of them to stabilize the molecule. But it's weak enough to go ahead and allow for this unwinding and unzipping so that we can make copies of the DNA molecule for transcription and DNA replication. Okay? So once again, the DNA molecule is structured in what they call a double helix, okay, a nice little spiral staircase. We have nucleotides that build it up, that's the monomer. So that's a sugar, phosphate, and base. And the bases bind together in a particular pattern with adenine always going to thymine and cytosine always going to guanine. Okay, this is according to Chargaff's rule, which is really cool because then if you know how much adenine you have, you can predict how much thymine you have. If you know how much of each of these, you know how much is left, and you can predict how much cytosine and guanine are left. So by knowing just 1% in terms of the bases, you can figure out the other three as well. It's pretty cool. Chargaff's rule is a pretty cool predicting tool. So again, nucleotide, sugar, phosphate, and base. Bases are bound together by hydrogen bonds. How they fit together is based on the number of hydrogen bonds and the shape of the actual base. Now, when we say nitrogenous base, that's because it's primarily it's got a lot of nitrogen in it. And so nitrogenous simply means has nitrogen in it. In DNA, the sugar is deoxyribose, but if we were to have a single-stranded version of this, and it's RNA, then it would just be ribose. But you would still have bases, and you still have your phosphate group. Difference would be is instead of ATCG, in RNA, you have a U, uracil replaces thymine in terms of our bases for a nucleic acid of RNA. All right, guys, have a good one. We'll talk to you soon. Bye.